Alrighty. Hello, everyone, and welcome to Pocket Size Bio with Dr. Casco. In this video, we will continue on the Campbell Biology textbook. This is Chapter 2, the chemical context of life. Right, the key concepts you can see here is what we're going to talk about. Matter consists of chemical elements in pure form and in combinations called compounds. An element's properties depends on the structure of its atoms, the formation and function of molecules, and ionic compounds depend on chemical bonding between atoms, and chemical reactions may make and break chemical bonds. All right, the chapter gives this visual here of wood ants, right? Formica rufa use chemistry to ward off enemies, right? When threatened from above, they shoot volleys of formic acid from their abdomens into the air. The acid bombards and stings potential predators such as hungry birds, right? Very nice little visual here. What determines the properties of a compound such as formic acid, right? We need to understand a little bit of what a compound is, right? It consists of several atoms joined in fixed ratios, right? Different types of atoms, right? We know them as elements. All consist having several properties, right? Number of protons, electrons, and so forth. Okay, so we're going to take a look at that today. So the first concept, 2.1, matter consists of chemical elements in pure form and in combinations called compounds, right? So this chapter is pretty much review of high school biology and chemistry class, right? So I won't go into incredible detail with the concepts, but I'll pass through every notable thing in the chapter. Okay, so to begin off, organisms are composed of matter. This whole physical world is composed of matter, right? And matter is anything that takes up space and has mass, right? And could, as we all visually see, has a variety of forms. Okay, but ultimately, any type of matter is made up of elements, right? And these elements are interacting together in different forms to create compounds. So I'm going to go a little bit up here. Okay. So, oops, little typo there. So matter is made up of elements, right? An element is a substance that cannot be broken down to any other type of substance by chemical reactions, right? It contains a particular property, right? There's 92 naturally occurring elements, and they're often designated by chemical symbols, as I will talk a little bit later, right? And then when you get several elements, you create a compound, right? Compounds are basically substances that consist of two or more different elements in a fixed ratio. That's a very important thing to know, right? A fixed ratio. And a very famous example is sodium chloride, right? Table salt. You have an element called sodium and an element called chlorine, and they react together in a 1-to-1 ratio. For every one sodium, you have one chloride atom. Okay. So that's what I'm referring to as a ratio, 1-to-1. Sodium, interestingly, as elements interact with other elements, they are able to have different properties, right? Table salt, a relatively um, safe thing to ingest, right? To eat, everyone eats it all the time. Table salts, to certain degrees, right? But sodium in itself is a metal, highly explosive metal, right? Chlorine is a gas in its own form, very poisonous, but when they react together, they create this very stable molecule, compound that is easy enough to eat and safe enough to eat, right? So whenever you have elements interacting or atoms of a particular element interacting, they can produce new properties or emergent properties, as we talked about in chapter 1. Okay. So again, going back to this idea of elements, right? The elements of life, since we're talking about biology. Of those 92 naturally occurring elements, around 20 to 25% are essential elements for living organisms in order for them to function properly, right? For the organism to be healthy and for all the molecular machinery pretty much to work well, right? We need 20 to 25% of these. Okay. And one kind of similarity among organisms is that we share these, we all need these similar elements, right? Or these same elements. Humans need around 25 elements. Plants need around 17. So it's not much really. 96% of living matter is made up of just these elements that you see here. Oxygen, carbon, hydrogen, and nitrogen, right? You should already be familiar with these symbols. Okay. Then you have these other, and let me just zoom out a little bit. Then you have these other elements, right, that you just need in minute quantities, right, or little micro quantities. They're called trace elements. Some examples are iron, right, iodine, okay, necessary for, again, functioning of these systems, biological systems. Okay. Iodine, in particular, if you have a deficiency of iron, you can have a goiter in the thyroid gland. So very important, despite the low amount that we need in the body, they're still critical for our normal development and maintenance of the body. The table you see here is an example of these elements that are necessary for a living organism. Okay. Again, like as I mentioned before, oxygen, carbon, hydrogen, nitrogen, right? They have a particular percentage in the body, oxygen being 65%, right? Carbon being 18%, and so forth and so forth. We also have these other trace elements, okay? Calcium, phosphorus, potassium, sulfur, sodium. It's good to be familiar with these. We will see them in great detail in terms of their importance throughout the chapters. Okay, sodium, potassium used for sodium potassium pumps for electrical impulses, right, and so forth. Okay, so again, some naturally occurring elements are toxic to organisms. Arsenic is a naturally occurring element in some areas, right, of the world, and it seeps into groundwater. Then you have arsenic laden water, right? Very, very poisonous to the body. All right. Just to give you another example here, a more specific example, which is quite interesting. Some organisms tend to adapt to these toxic elements, right? Serpentine plant communities, right? So you have this area in a peninsula that's around San Francisco Bay where you have this mineral, serpentine, right, that contains chromium, nickel, and cobalt that is quite toxic to living organisms. But some plants have evolved and adapted to thrive in these type of conditions where you have this mineral, right? Most plants cannot survive in this area, in this type of soil that has this mineral, right? So that's quite interesting there. Evolution, adaptability of certain organisms to thrive in a particular environment. Okay. They're referred to as serpentine plants. Okay. Just to give you a little concept connection check that you can kind of self-assess yourself there. Okay. We're going to go on to concept 2.2. All right. I do apologize if you hear some construction in the background. Hopefully not, but they're kind of renovating the facilities here. All right, so an element properties depend on the structure of its atoms. Again, this is review for a lot of us, right? An atom in itself, what is an atom, right? It's the smallest unit of matter that still retains the properties of that element. Generally, we will use symbols to denote this particular element. Usually, it's one capital letter some elements have a lowercase a second lowercase letter, right? So you generally use symbols to denote, right, the elements. C for carbon, H for hydrogen, Fe for iron, right, and so forth and so forth. An atom in itself is composed of a couple of subatomic particles, neutrons, protons, and electrons. Their name gives that characteristic charge that they have. Neutrons have no charge, protons have a positive charge, electrons have a negative charge. Okay, there's several simplified models we're going to see throughout the textbook on how an atom is portrayed. This is one example of a electron model. Here's a cloud, right, of electrons that is surrounding a nucleus of an atom having protons and neutrons, right? This is another simplified model that you see. You can more formally see an electron shell where you have the likelihood of electrons being found. Okay, so there's different ways that you can represent this, and we're going to see several. Okay, like as I mentioned before, you have these subatomic particles. The nucleus is basically that central core of an atom, and you'll find the protons and the neutrons aggregated in this core there. So you see the central region there. And then electrons are pretty much moving in an orbit, right, around this nuclei, right, and around this nucleus. Okay. When you refer to the neutrons and protons, right, they're the only ones, if I can say, that have a notable mass to be measured. They're large enough that we can detect a mass, right? Electrons are um, they're so light that it's almost negligible, right, to any reading. So um, we really don't take it in consideration when we're measuring masses, right, of an atom. In particular, you would measure neutrons and protons, and neutrons and protons they're almost identical in mass. Okay, and they are essentially, 1.7 times 10 um, uh, to the 24 uh, grams. And it's not so useful to write it in this way when you want to calculate things. So, um, John Dalton, right, a physicist, a biologist, a chemist, developed the atomic theory, and one of his kind of postulates on this. Postulates is maybe a strong word. One of the things he pretty much established is the atomic mass unit, right, which is referred to as the Dalton, right? So protons and neutrons have masses that are close to 1 Dalton, right, or 1 AMU. And with this, we can establish several things or several properties of the atom. Okay, one of them being the mass number, right? And the mass number is basically the addition of protons and neutrons, right? In the case of sodium, and you see here in this little diagram, sodium has a mass number of 23. Okay, and generally you'll designate the mass number in this upper kind of superscript area of the symbol, right? And then you have the atomic number, and the atomic number defines what the element is, right? And it's characterized by the number of protons only. Okay, the number of protons um, uh, you would find in the subscript of this sodium symbol here. And in this case, it's 11. Every atom of sodium will have 11 protons. Okay, and in a neutral atom, you'll have 11 electrons also. Okay, so generally in this kind of ideal element, you would have um, the number of neutrons would, uh, if I can say, they'll equate to the number of protons, but that's not always the case. So if you have the mass number and you want to calculate the number of neutrons, you can just subtract, the mass number minus the atomic number, and that can give you the number of neutrons. Okay, but as we see, as we're going to see with isotopes, right, it's not always one for one, the same number of protons with the same number of neutrons. That could change, as we're going to see in a little bit. Okay, so uh, yes, atomic number is a generally fixed number. Is a fixed number. Mass number could change, right? But all in all, in like conventional basic periodic tables, you have that fixed mass number, right? 23. Um, then you have something called atomic mass, which is much more defined. It does there is a slight difference between a mass number and atomic mass. Okay. Mass number is just the addition of protons and neutrons. Atomic mass is a kind of average of the different forms of an atom, okay, based on the number of neutrons that it contains. And these are referred to as isotopes, right? So if you see this little visual here, you have carbon 13, you have carbon 12, carbon 14, right? They all have the same number of protons, but they have different number of neutrons. So that means that they'll have a different mass. They'll have a different mass number. So an atomic mass is essentially the average of all the different forms of carbons of these isotopes, right? The scientists calculate with their relative abundance on Earth. So what would be let's say 23 for sodium could be 22.987, 6666, right? Depending on the relative abundance of these different forms of sodiums. And the same thing goes for carbon also, right? And all the elements. Okay, so make sure you keep that in mind. Mass number is not necessarily the same as atomic, is not the same as atomic mass. Okay. And again, these different forms, when you're referring to masses, right, and different numbers of neutrons, they're referred to as isotopes. Okay. And you can have different forms, not just of carbon, you have uranium, nitrogen, and so forth. But one thing to keep in mind is that they still can perform the same chemical reaction. Their ability to react doesn't change. Okay. Okay. There's some types of we're going to see this later, but there's some types of isotopes that when they have a too much of a high number of neutrons, they become radioactive. Means that they're a little bit unstable. So they tend to release a lot of energy and give off particles as they're decaying, as their nuclei decays. Okay. And there's many applications in biology and in science in general, in terms of radioactivity that we're going to take a look at later. So radioactive tracers, right? So, yes, radioactive isotopes, right? They're highly used in medicine. Okay. Cells assimilate these atoms just like stable forms, so we can absorb isotopes just like normal, the most stable forms of these atoms. Okay. And these radioactive forms can be tracked, right? Visually through, imaging, different types of imaging. And you can see where a certain type of tissue is, right? For example, cancerous throat tissue, as you see here, right? Due to certain properties of some biochemical pathways, you can use a particular target molecule or target element, right? And if it's stuck to a particular thing, a particular tissue, you can use that as a form of diagnosis. Okay, so some examples are kidney diseases, PET scanners, right, to view images of tissue, elevated glucose, or measuring just cancer development. Okay. The use of radioactive material has to be taken with care, right? Definitely there are threats, right? Overexposure to human health, right? And the environment. As we're going to see much later on, radioactive material is quite damaging to cells and molecules. So you do have to be careful of that. Okay. Another kind of application is radiometric dating, right? It does deal a lot with evolution and how old certain material are, like fossils, um, throughout life. Okay, but scientists can apply this idea of radioactive atoms to date fossils through a concept called radioactive decay. Okay, so fossils, they provide a large body of evidence, right, to support evolutionary theory. And again, this is something we're going to take a look at later. And isotopes, when they're radioactive, they tend to, decay into different elements. Okay, so for example, carbon 14, and I'm going to zoom in a little bit here. So carbon 14 can break down over uh, um, a fixed time frame, right? Could decay into nitrogen 14. And as it breaks down, it liberates different particles and energy, right? So scientists are able to calculate this rate of decay. Okay, and that is referred to as radiometric dating. Okay, so just to kind of read the prompt here, the notes here, an isotope, sorry, an isotope decays into daughter isotopes at a fixed rate, right? And this fixed rate is usually referred to as half-life of the isotope. Okay, when it decays, it usually when it reaches this half-life or this fixed decay rate, right? 50% of that parent isotope would break down into this other form, right? So it's very specific to an element, right? Carbon 14 has a very specific decay rate, right? Or half-life. Uranium has its own decay rate, right? And so forth. So you can use this to figure out how old a fossil is. Okay, so radiometric dating measures a ratio of different isotopes and could calculate how many half-lives have occurred, right? Um, since an organism, was alive to the point that you discovered, that fossil of that past organism. Okay. Again, time ranges are could range between elements, and you can go for a couple of thousands of years to millions of millions of years. Okay, to billions even. Okay, as I mentioned before, so uranium 238, which decays, has a very very long half-life. So scientists can use that to measure the age of the Earth, right? 4.5 billion years ago. Old, sorry. Okay. And again, we get into this a lot more in evolution. Okay, these are nice scientific skills exercises. I do recommend you guys to do. Um, but yeah, if you have any questions on that, you can leave a comment or let me know. Okay. Continuing on 2.2, right? You have the energy levels of electrons, right? So this is moreover talking about the conformation of how electrons are organized within the atom. Okay, just to give you some representation of the core of an atom and the distance of an electron. You see this commonly in textbooks, right? The representation of an atom is more like a fruit fly around a stadium, right? With the nucleus at the center, right? So you have the nucleus at the center of a field, and then you have a little fruit fly going around, let's say in the seats, right? The seat areas. So the atom is mostly empty space, right? And um, when it comes to chemical reactions, the only ones that are really interacting between different atoms are electrons. Um, Not even different atoms, same atoms also, carbon to carbon, right? So electrons, they can have different levels of energy. Right? We know that energy is the capacity to cause change or to do work, right? And potential energy is energy that matter has due to a particular location, right? Structure, condition. So, having that said, electrons have certain levels of energy depending on where they are in relative to the nucleus of an atom. Okay. Matter in general has a natural tendency to move toward lower energy states. Okay. And I'm going to explain this with the visual we have here. Electrons have potential energy due to their distance of the nucleus. So, in order for an electron to move to a different energy level, right, or shell, and these are locations where electrons are most likely to be found, right? You need a certain amount of energy, right? So again, make sure you have you note this, right? Electrons exist in fixed energy levels or shells. Okay, and that energy level correlates with the distance with the core of the nucleus of an atom or the nucleus. They're usually represented by circles, right? As you see here. So you have the atom, you have the nucleus, you have that first energy level or the first shell, you could also call it. And then you have a second energy level and you have a third energy level, right? And as you're going down to different energy levels, you tend to lose energy, right? But if you're gaining energy, okay, you kind of excite an electron that it can actually jump from one level to another, as we're going to take a look um, at that later. So the first shell or the first energy level has the lowest amount of energy. Okay. And then as you go outward, there's more energy within the shells. Okay. And electrons can bounce between these energy levels depending on how much energy they gain or lose, right? Okay. So in terms of electron distribution and chemical properties toward chemical reactions, right, the chemical behavior of atoms, okay, is determined by the number of electrons on the energy levels, right, on the electron shells. Most in particular, the outermost shell. Okay. Just to give you a representation here of hydrogen. Hydrogen is the simplest element, and you see here it only has one energy level. Of course, hydrogen has an atomic number of one. It has one proton, right? So it generally would have one electron, right? And that electron would be in this first little electron shell. Okay. So just to zoom out a little bit here. So in this little visual here, this is a modified little periodic table here. As you're reading from left to right, you're seeing there's an increase in atomic number, right? There's an increase in electrons that can be found in the electron shells. For example, helium having two protons has two electrons. And then you see that there's these two electrons in the electron shell. Okay. Some characteristics to note that the first shell can only hold two electrons. Okay. And the only elements that have one shell is essentially hydrogen and helium. Okay. And then the first shell can only hold two electrons. If you have more electrons, you need to have the next level shell, right? The second shell. And then here you can see this row of elements that they all have two shells, right? The first shell and then the second shell. And then you can start filling in the electrons in that second shell. Okay. The second shell can hold up to 8 electrons. Okay. And then this is an important thing to note here. Okay. Chemical behavior depends mostly on the outermost shell. And that outermost shell is also called the valence shell. And that's where you're going to find the valence electrons, the number of electrons on the outermost shell. And those are the ones that undergo chemical reactions. Okay. A characteristic about these elements that have a similar number of valence electrons means that they generally exhibit similar behavior. Okay. And we'll get into that also in a bit. Okay. All right. A little bit more on kind of configuration of electron shells. You have electron orbitals, right? So as we see here in these models, right, electron shells are represented by, these little circles, okay? But a more accurate depiction of these electron shells are referred to as orbitals, right? And they're representations in a 3D space where an electron is most likely to be found 90% of the time, right? They're referred to as orbitals. Okay, now I'm going to go a little bit down here. Orbitals, they're often thought as components of electron shells. Okay, so if you were to see hydrogen, for example, right? Hydrogen just has that first electron shell. So more specifically in a 3D space, it would have this little orbital here, this little spherical orbital, okay, where the electron is floating around the nucleus. Okay. And if you have an atom, let's say lithium, right, that has three atoms, three um, uh, electrons, okay, just to kind of show you the visual here. So you have the first shell, second shell, okay. You would have a second shell that the second shell itself would have a orbital, right, spherical orbital, and then it would have these little dumbbell forms of orbitals, right, depending if you have more and more and more electrons, right. These little spaces, these orbitals, they have particular names, right? They're referred to as 1s or one spherical, a spherical orbital, right? That's the first shell. The second shell has four orbitals, one large s orbital. It's referred to as 2s orbital, right? And then it has three dumbbell-shaped orbitals, as you can see here. That's one, right, on the y-axis, on the z-axis, and on the x-axis. Okay, and these are referred to as the three 2p orbitals, or 2p orbitals. Sorry, they have three dumbbells. Okay, x, y, and z axis. Okay, in any orbital, one thing you do need to know: in any orbital, you can't have more than two electrons. Okay, when you have two electrons, you fill that orbital there. So again, reactivity of an atom depends on the presence of an unpaired electron. Right? So if you have one orbital that is having only one electron, then it has potential to react with another atom. Right? Again, we're going to see this in a little bit. So just to show you a little final representation there. Okay. So you have your first electron shell, the spherical orbital. Then you have your second shell, which has another spherical orbital and the dumbbell orbitals, the p orbitals. Okay. Within biology, you don't get into that much level of detail with electron configurations, so you don't need to worry so much about going further than what you see here. Okay. All right. Oh, a little bit more in terms of chemical reactions and bonding. So this is concept 2.3. The formation and the function of molecules and ionic compounds depend on chemical bonding between atoms. So let's take a look. So atoms with incomplete valence shells, it can interact with other atoms in such a way that each partner atom completes its valence shell. atoms like to have their shells full, right? The way they do that is by chemical bonds. And there's two ways that you can fill your electron shell, right? You can either share electrons or you can transfer electrons from yourself to somebody else, or you can capture an electron from somebody, from another atom. Okay. So that interaction in itself of sharing electrons or transferring electrons is referred to as a chemical bond, right? The strongest type of bonds are covalent bonds and ionic bonds, and there are different types of bonds that we're going to take a look at, especially and referring to ionic bonds, especially for dry compounds, right? So in terms of covalent bonds, right? Covalent bonding, you have this situation where there's a sharing of valence electrons, okay, by two atoms. For example, hydrogen and hydrogen bonds, right? So remember, they have 1s orbital. So let's take a look here, right? And since one hydrogen atom has one electron and the other one has one electron, and remember, for the first shell, you can hold only two electrons max, right? So in order for them to fill both of them that electron shell, they share their electrons to each other, and now they both can fill their orbital and they're sharing electrons, right? So this would be a form of covalent bonding. Okay? And this would be more specifically something called a single bond. Okay, you can actually share more than one electron, right? Here you have an oxygen molecule, right, where you have one oxygen that is bonding to another oxygen, and here they're sharing four electrons with each other, right? And here you refer to this as a double bond, right? Where you have two pairs of electrons being shared amongst these two atoms. Okay. You don't necessarily have to have the same atom. You can also create a covalent bond with different types of atoms. Water, very famous example. You have hydrogen bonding with an oxygen atom, right? And then you have another hydrogen bonding with that same oxygen. Then they're sharing one pair of electrons. Ultimately, everyone has their valence shell filled. Okay. Let me just go a little bit back to the notes here. Okay. When you have two or more atoms held together by covalent bonds, you will refer to that as a molecule, generally, right? And what I was showing you previously, right now, is a Lewis dot structure, right, or structural formula. Also, we see here, and a space-filled model. You'll see these different forms throughout the textbook many times. So it's good to familiarize yourself with that, with a modeling convention, right? These are Lewis dot structures. This is a structural formula. Oh, sorry, this is an electron distribution diagram. This is a Lewis dot structure, and this is a space-filled model. Sorry about that. Okay. Let me just zoom out again. All right. So a term related to this, the bonding capacity that an atom has is referred to as an atom's valence, right? Some molecules, they're diatomic molecules like hydrogen, H2 and O2. They're referred to as pure elements rather than compounds, right? Compounds is usually a combination of two or more different elements. Okay. So make sure you have that. You note that. Okay. And atoms that are sharing electrons, right? This is another property that we're going to see a lot more when we talk about biomolecules, right? Atoms that share electrons can pool an electron, right? Unequally, right? Doesn't you don't necessarily have an equal spacing between one atom and the other. Sometimes one atom pulls it more towards itself than the other, right? Or to a certain degree, right? This idea of unequal sharing, right? Because one atom is pulling more than the other, is referred to as electronegativity, right? Some atoms are very electronegative. And as you increase electronegativity, you're going to pull electrons toward yourself, toward that respective atom. Okay. Um, Oxygen is a very electronegative atom, so it tends to pull electrons toward itself. A very, very famous example is water. We're going to see this in a lot more detail later on. So hydrogen is not electronegative, right? Um, at all, if I can say. So oxygen will pull the electrons, or I want to. It's better to refer to it as oxygen being more electronegative, so it's pulling, right, uh, the electrons toward itself. So electrons are passing to oxygen. They're still sharing, by the way. I want to focus on that. They're still sharing the electron, but it's just more accumulated to one particular area. If I were to put, let's say, a little dot here. Let's say the electrons would be more like that. Selfish oxygen, right? So because of this unequal distribution, you're going to have hydrogen and oxygen having a partial charge, right? Or some little form of charge where you would say oxygen is partially negative and hydrogen is partial positive. Okay? They have a little charge there of positivity here and electronegativity here. Okay. They're referred to as partial charges. Any molecule that has this situation occurring are referred to as being polar, right? And when you have this unequal distribution, you refer to that as a polar covalent bond, right? Because of that unequal distribution of electron. When you have an equal distribution, let's say two oxygen molecules, atoms that are sharing electrons equally, since they both have that equal pool, you refer to that as being nonpolar. Okay? There's an equal sharing. But whenever you have an unequal distribution, it's referred to as polar, right? A polar covalent bond. Okay? And you would refer to these atoms as being partial positive and partial negative. And they're usually um, uh, you use the sigma, right, symbol to designate the charge, right? Partial negative, partial positive. We're going to see this later. This creates another type of bond called hydrogen bonding that is critical for in biology. So we're going to take a look at this later on. Okay, let's continue on. Ionic bonds. Nothing really extraordinarily new to us. We should be familiar with this already from physical science in middle school, right? When you have an extremely unequal distribution of electrons, so much so that one atom will essentially take away the electron from the other atom. And that in itself will create an ionic bond, right? Due to the formation of what are called ions. Ions is basically an atom that has lost or gained an electron, so it produces a charge. Any ion that has that has gained an electron will have a net negative charge, and it's referred to as an anion, right? Any atom that has lost an electron would have a net positive charge, right? Because you have more positive more protons than electrons, you would have a cation. Okay, so if you see in this visual here, right, sodium will lose an electron to chlorine. Both of them, again, have their shells filled very nicely. So now sodium has a positive charge, chlorine has a negative charge. Sorry if you can't see the visual there. Um, and the resulting formation is an ionic bond, sodium chloride. Okay, positive and negative. Let me just go a little bit down here. So you'll generally have compounds, right, formed from ionic bonds, and they're referred to as ionic compounds or commonly known as salts. Okay, again, sodium chloride being the common one. And in their 3D structure, they generally form crystals. Okay, and these crystals can grow and grow and grow, so they don't have any fixed shape as you would see in covalent bonds, like biological molecules like the cell membrane or cholesterol, for example, right? So they're usually found as crystals, right? And these crystals can vary in sizes and shapes depending on the properties of this of the lattice and of the atoms. Okay, so you have this 3D lattice that you see here, or this 3D mesh. Okay. Again, like I mentioned before, ionic compounds don't have a definite size or number of atoms. They can continuously add on and add on more, right? So in that sense, they're different from covalent compounds. Okay. They tend to have a fixed ratio though, right? For one sodium, you'll have one chloride. Okay, sodium chloride. You could have magnesium chloride. For one magnesium atom, you have two chloride ions, and so forth. Okay. The term ion doesn't necessarily have to do just with a single atom. You could also use it for a molecule. For example, like ammonium chloride, NH4Cl, which will produce, you have this NH4 plus, right? This ammonia ion and this chloride ion. Okay. It doesn't have to be just one single atom. It could be a little compound of NH4. All right. And related to that is the environment affects the strength of ionic compounds. Generally, so as you have a drier environment, that will produce a higher bonding strength because when you have water in the vicinity, water will tend to dissociate. Okay, will weaken the bond and just dissociate and break apart the ionic compound. So the drier the environment, the stronger the bond strength between the two ions. Okay. Additional to that are some other interactions, right? Hydrogen bonding, van der Waal interactions. We will talk about this later on, especially hydrogen bonds. But they're referred to as weaker interactions that are within the molecule and even in between different molecules, critical to life. You might know already just the idea of DNA, right? And the two strands of DNA and the nitrogenous bases, hydrogen bonding to each other, right? So again, not only covalent ionic bonds, but you have these interactions which will produce more emergent properties of life that we'll go into detail later, right? Good. So hydrogen bonding, right? It's a uh, another form of weak interaction. Okay. Uh, very, very critical to the chemistry of life. Again, this is formed from the idea of polar covalent bonding of a water molecule, right? The partial positive atoms of the hydrogen would create interaction, a hydrogen bond with a partial negative atom, okay? A water with water molecule or a water with ammonia molecule, right? This is another polar molecule. And if you see here, right, you have the hydrogens kind of being attracted to the negative nitrogen, and it'll create this interaction here, this attraction of partial charges. Okay. And that interaction, that attraction between partial positive and partial negative is referred to as a hydrogen bond. Relatively weak bonds, right? But when you accumulate many and many of them, it starts to give some very interesting emergent properties, right? Depending on the situation. Okay. So that's hydrogen bonding. Van der Waals interactions, right? Another type of attraction, right? and and even when you don't have polarity, right? This can occur with non-polar covalent bonds, right? They can have some partial, some negative and positive regions, right? But uh, as a net, right, they have no charge. These non-polar covalent bonds. So electrons at any instant may not be evenly distributed within the molecule, right? So that gives a chance for one area of the molecule to have a certain amount of charge, right? So it's a relatively weak interaction, right? Van der Waals interactions are individually weak and occur when atoms are very, very close together. Okay. An example of this is the gecko, right? With its appendages, right? Its little fingers there, and these interactions between um, the little hairs of the gecko onto the surface of whatever it is attached to, right? Creates this attraction, right? That enables it to kind of stick to a surface. And of course, having more, if I remember septa, right? Uh, you maximize surface area, so more attraction, more adhesion could occur, right? In the case of the gecko. Okay. These interactions can happen of regions of large molecules like proteins and nucleic acids that we're going to take a look at also. Okay. Very, very characteristics is molecular shape and function, right? So depending on how you're constituted, right, the elements that you're using, the atoms that are building particular molecules, you will have a characteristic size and shape, right? And that's key to the functioning of a cell. Okay. So shape is very, very important. And different molecules can have different types of shape depending on their orbitals, right? Depending on their orbitals and depending on their bonding, they can have particular 3D structures. Okay. Sometimes you can have linear shapes like O2. If I'm not, no, I don't have an example here of oxygen. Okay. So the molecular shapes that these different molecules have is dependent on the orbitals that you have. Okay. So if you have atoms that have s and p orbitals and you have single bonds occurring, you have a 3D formation called a tetrahedron. Okay. Let me just go down a little bit ahead. Methane, for example, CH4, right? You have this molecule that has one carbon that is creating single bonds to four different hydrogens. Okay. So it's creating this kind of tetrahedral shape, this little pyramid shape. Okay. Just as you can see here. All right. And then you can have another type of bonding. Okay. Like water. Water has a bent structure because it has a little bit of unbonded pairs or lone pairs of electrons, right? So you have these orbitals here that are not being used. So because of that, you have this kind of bent structure here, this little V structure. You have oxygen, hydrogen, and hydrogen, and they're bent on this particular angle here. Okay. So by having this idea that orbitals can create a different shape and things, when you have many, many molecules in different areas of a protein, you can create very complicated forms, very complex forms of proteins in particular, right? Let me just go to this particular part here. Okay. So molecular shape is crucial. This is a very, very interesting part in biochemistry. Okay, so molecular shape, as I mentioned before, is critical. It determines the how a biological molecule works, right? And we're going to see this a little bit later that how a molecule is built, it can recognize and respond to some other molecule with incredible precision and create a chemical reaction, create an effect in the cell. Okay. And bonding, whenever they're interacting together, these two different molecules, they could be temporarily, and you can have complement shapes, right? Similar to kind of a lock and key model of an enzyme and substrate. Okay. You have an example they show here, right? Morphine and heroin that is derived from opium. Okay. And if you take a look here, let me just zoom out a little bit so you can see better. Okay. Referred to as molecular mimics, right? Morphine is a a medication that helps kind of ease and relieve pain and mood, and it's used in patients a lot to kind of basically a painkiller, right? So you have these opiates that in general they relieve pain. They also alter mood, right? And they bind weakly to receptors on brain cells, right? Now the thing about this is that, how can brain cells have receptors that bind to opiates made from plants that are not endogenous to the body, right? So endogenous is a critical word, right? They're made from the body. They're native to the body. We produce it naturally, right? So um, endorphins, right? Endogenous morphines, they're naturally made signaling molecules. They're made in the pituitary gland. They generally relieve pain, right? And you generally have an euphoria they produce euphoria during stress. So you can have medications like morphine that have a very similar chemical structure. Okay. And because they have a very similar chemical structure, they can bind to the same receptor of the brain cell to create that pain relief. And that's the whole idea of medicine, right? Creating or finding other chemical compounds that are very similar that could have a similar effect, right? And this is a whole area of science, of biology, of medicine, um, that deals with drug discovery. It's a good hotspot if you're interested in doing research. You can do in silico methods, computational methods in finding different types of drugs for a particular disease, right? Just by computational methods, right? It's referred to as drug discovery. Um, Especially now with AI, it has created a lot of breakthroughs in drug discovery. All right, so this is very critical in terms of molecular shapes. Okay. All right. And let's go to this last portion of chapter 2, which is chemical reaction. Again, these are things that we've seen before in middle school and in high school biology. Chemical reactions make and break bonds, right? So, um, making and breaking chemical bonds will ultimately lead to changes in the composition of matter. Every time you have the formation or the breaking of chemical bonds, one way or another, it's a chemical reaction, right? So you have reactants in this chemical reaction, the molecules and compounds that are interacting together to create an output. And that output is referred to as the products. So in this case, let's say you have two molecules of hydrogen and a molecule of molecular oxygen. They react together to produce two molecules of water, of H2O. Okay. So a little bit of kind of writing, you have coefficients. Coefficients are these large numbers. They're referred to as the number of molecules that you have um, of that particular molecule. So in this case, I have two molecules of H2. Here, you just have one. One is generally not written, right? And the same idea goes. You have two molecules of water. Okay. Generally, when you write these chemical reactions down, you have to balance the equation because of conservation of mass. You cannot create matter or destroy matter. It just is transformed into two different outputs, different molecules. Okay, so matter must be conserved in chemical reactions. That's a little bit beyond the scope of biology in terms of balancing equations. You'll see that in chemistry, right? Um, a famous example is photosynthesis, right? Takes place in plant tissues. We're all familiar with this process. Humans depend on this process for food and oxygen, not just humans, but life in general. It's the foundation of life, um, and for almost all ecosystems, right? So you have 6 molecules of carbon dioxide, 6 molecules of water through various reactions, right? This is a very simplified form of the reactions, but ultimately you get carbon dioxide and water, um, driven by sunlight, right? You have an output of products of glucose, C6H12O6, plus 6 molecules of oxygen. Okay, if you count the number of atoms, you'll see that the equation is balanced. Okay. One thing to note about chemical reactions is that, in theory, all chemical reactions can revert back, but it just depends on number of collisions, concentrations of the reactions, the amount of energy that you require to revert the reaction, right? Uh, but it is possible to revert reactions, or to be reversible. Another example here, you have 3 molecules of hydrogen plus nitrogen could create this ammonium molecule. Okay, and revert back. They're very important in terms of understanding equilibrium and changes in chemical reactions and overall net flow of uh, uh, of product to reactant ratios. Okay, if there are more reactants, then the reaction goes forward. If there are more products, then the reaction could reverse. Okay, there is a equilibrium, a chemical equilibrium that is occurring. Okay, so you can say that the reactions are dynamic. Reactions are still occurring, but there's a net change, right? Um, between the products and the reactants. Okay, and this is 2.4 for you guys to see here. And with that, we finished